Well, hello, everyone.
Yes.
So welcome to Spring 2023, the GG Seminars. And it's my real pleasure to introduce Adam Rosenthal. He just started his lab. He's assistant professor in the Department of Microbiology and Immunology at UNC Chapel Hill. And I've known Adam for at least 10 years. We were both on a large grant through DARPA, which, if you've ever done it, is really crazy. There's a lot of quarterly reports and a lot of wild ideas out there between different groups and so on. And it was a real pleasure meeting Adam, who at that time was a postdoc from Caltech, and we both had shared interest in looking at microbial organisms, but at single cell. And you'll hear much more about this today from Adam. Just a brief bio on Adam. He got his PhD in molecular biology at UC Los Angeles, where he worked in the lab of Jay Gralla. For those of you who know Jay Gralla, it's a mechanistic lab, and he worked on mechanisms of regulation of osmotic stress response in the classic bacterium E. coli. He then did a postdoc, moved 30 miles away, I guess, to Pasadena, where he did a postdoc at Caltech in not one, but two different microbiology labs. One was in the Ledbetter lab, where he looked at termite gut microbes, so spirochetes, and sort of the hydrogen economy that's involved there in the termite gut. And he also worked on a classic model organism thinking about cell fates and thinking about single cells, Bacillus subtilis in the Elowitz lab, where he looked at metabolic partitioning. He then moved to, if you haven't heard of this, Calico Life Sciences, which was an initiative started to study aging. And so this was in the Bay Area, and he was a group leader there from 2015 to 2017. And then he moved to DuPont Nutrition and Health, where he was a senior principal investigator. And I think you'll hear about some of the technologies he's been developing, like he has been developing throughout his postdoc, but also as a group leader in these industry positions, so like Dropseq and bacteria. And so he's been an assistant professor since July of 2022, and he'll be telling us what he's been up to and where he's going. Welcome, Adam.
It's a pleasure to be here. Really happy to be back in academic settings after a long hiatus. It's really fun. I can talk about all the research. Even more fun to be, right across the street and meet new colleagues and neighbors, and hopefully lots of fruitful interactions. So I'm really, really happy to be here, having a great time. I'm going to talk today about some work, as Nick mentioned, from my postdoc with Michael Elowitz, and then about some new things. But really centers around the idea of single cell analysis and heterogeneity in bacterial populations. And so in the last few years, we've heard a lot about single cell analysis in mammalian systems. And it's no surprise because we think about mammalian systems as multicellular organisms. And so in the human body, of course, we have different cell types that carry out different functions. Now, it would look very different if I had no bones. I wouldn't move around if I had no muscle. My immune system would be very different if I didn't have different types of immune cells in it. And so really we accept that there are multiple cell types that work together in a genetically identical background host when it comes to higher life forms, if you will. But this talking about bacterial communities. In bacterial communities, I don't think I need to convince people here, especially folks working on environmental organisms, that multi-species bacterial communities are really well organized, and you have different specialties and niches and different cell types that work together for the good of the entire community. But really what I'm going to focus on today is on single species bacterial communities. Clonal populations of bacteria and how much heterogeneity exists within those populations, right? And so we've known that bacteria can form specialized cell types when grown in isolation from the dawn of microscopy. What I'm going to talk today about is that these specialized cells play an important role in the disease industry and the environment, but why do they exist? And what's the function and how are they regulated is really some of the questions that we're thinking of. So I'm going to cut this into three parts. The first one is going to look at a study looking at Bacillus subtilis using traditional single cell analysis tools, fluorescent reporters, in movies, and trying to understand the regulation and maybe some of the roles that this heterogeneity plays. The second part is a new method that I developed while at DuPont, which I think is very powerful. And I sort of want to introduce to you guys. And then the third part is, okay, well, showed us that there's a new method and that there's really interesting biology. Well, what's open now? What can we actually answer? All right. So I mentioned that we have seen cellular heterogeneity and specialization in bacteria for a really long time. Here's a picture from the Botanical Gazette in 1892, taken by Roland Thaxter, who was a botanist in Harvard, but this is not a plant. This is myxobacteria. And back in those days when you didn't have great cameras, you had to be an artist as well. He was also a pretty good artist. Of course, looking at this structure, we can speculate and would be correct that spores live on top of these structures. And we can say, okay, well, maybe there's an actual advantage to this, just like the bones I talked about earlier, that the spores are on top and they can be dispersed further. And so in some ways, this single-celled organism actually does have function much like you would otherwise. So that's a great example. What about heterogeneity that's harder to see, though? How much of that exists? And what are the roles that actually carries out? Is it purely like sporulation, or are there other things? When I was in the Elowitz lab, we studied Bacillus subtilis exclusively. And one of the nice things about Bacillus is that it sporulates just like the myxobacteria, except it doesn't make quite as beautiful structures. At least arguably it doesn't. But it also has other cellular behaviors that are heterogeneous. This is genetic competence, the ability to take up DNA from the surrounding. And so here's a picture from the Elowitz lab back in 2006, in which you can see that there are at least 3 cell types in this diagram. There are cells that have sporulated, there are cells that are basically competent, and there are cells that are neither. And if you look at it more closely, you might say, oh, this cell looks chunkier than this one, and maybe there's more heterogeneity. Don't know. And so what's the reason that we do this? And working in the Elowitz lab and with a lot of systems biologists, the way competence and sporulation are really thought about is they're thought about as bet hedging strategies. The idea is that these bacteria sort of think collectively and they say, hey, you know what? We're just simple bacteria, but as a community, we should maybe hedge our bets. Maybe conditions are going to be really bad, so some of us should sporulate and we'll have some progeny that survives these bad conditions, but maybe the conditions will get better. So for some of us to stay vegetative, we can exploit that. Competence is thought of the same way. Maybe there's some genes in here that are useful. If one of the bacteria takes up these genes and they're deleterious, okay, small fraction dies. If another, right? But bet hedging is really not the same as the multicellularity that you have in higher organisms, in the sense that it's forward thinking, meaning it's about the future conditions that you're betting may or may not happen. It's not about interactions in the moment. It's not like my muscles moving right now or my immune system. And so we thought maybe we can look at things that are more actively collaborating in real time. And to do that, we turn to metabolism. And so metabolism in multicellular communities really organizes the community. You have different niches. But these maps that we use are really a static representation. Does every cell do everything at any given time? Or some of these things go in one way or another? No, they don't. And so which cell types are active for particular pathways? How is it organized in time and space? And are there metabolic incompatibilities that actually regulate how the community is structured? Those are some of the questions that we want to ask. And what we did is we basically decided to start, if you're going to start making metabolism at central carbon metabolism, and we made a whole bunch of reporters for different genes in central carbon metabolism in Bacillus subtilis. So these are promoter fusions that basically drive GFP or YFP. And we see that even in TCA cycle genes, there are genes that are bimodally expressed. Okay. And we can see those throughout central carbon. And we can actually then look at the growth curve of the bacteria and try to see, okay, are metabolites also changing? Because more than just transcription, there are other mechanisms that we need to look at. And in fact, metabolites like acetate go up and then go down, even as the cells are growing, and pyruvate, et cetera. We wanted to see whether these populations that are associated with metabolism actually track. And they are a population that we ascribe as potentially acetate producing, comes up at the same time that acetate does, and one that produces acetoin, which is an uncharged metabolite that comes on later, comes up when the population does. If you guys have really good eyes, you might have noticed that this picture is actually from the same field of cells. If you overlay those two cells, you can see that it's really an either or. Cells are either in that acetate producing or the red population for gluconeogenesis. And if you look at more than one field, you see that this really is anti-correlated. And so these cells are not just expressing one gene on and off and it's random. It looks like some things are actually coordinated. So to look at the coordination of more than one gene at a time, back in those days, we just had fluorescent reporters. What we did is we fixed cells and then we sorted them, which in mammalian and I guess plant systems to some extent too. People don't do this much for bacteria. But when we did this, we found regulators and other genes. So this is competence regulator actually regulates a TCA cycle gene. And we can see that the same cells are on for both of these. And we see hundreds of genes as you would in RNA sequencing. Because we knew the regulator, we can actually knock out this population and see that we no longer make acetate as we predicted. So this is more than just transcriptional heterogeneity. This population actually has a metabolic role. So what I've showed you is that even in exponential growth, a subset of cells is metabolically different and produces acetate. We can start to look now at whether cells are stuck in this stage or not. We use a device called the mother machine in which media is flown and you have cells that are stuck in channels that are the length of one cell. And when you make this is that acetate producing gene, this is the TCA cycle gene and acetate production, you can see that there are cells that are basically going to switch on and off. These cells grow really slowly compared to other cells. You'll see another one up here, I believe. And so we can actually look at it, but cells will switch out of this state. Okay. The other gene, which comes on later for acetylcholine production, has a different dynamic. Cells are much faster in switching in and out. And we can take these movies and then analyze them in different ways. One way that's visually pleasing, you take one of these channels and basically superimpose different times of it. So now we're going to just look at time 1, 2, 3, et cetera. And we can look at how a cell actually switches in and out. And so these are two sister cells. One of them turn on the TCA cycle gene and basically grows slowly. And at this stage, it basically switches out. And there's the sister gene that didn't. You can see how many divisions happen in that cell. And so these cells that are growing in exponential phase are growing very, very differently. This is the one that produces acetylcholine. And I mentioned before that acetylcholine is basically something that the cell makes after acetate rises. And the reason is that acetate becomes toxic to the cell. In yeast, the same thing happens with ethanol production. So produce a lot of ethanol and then start to use it and secrete other metabolites. Acetylcholine is used this way by gram positives, or some gram positives, and it's thought to be a detoxification mechanism. So we thought maybe some of the cells are detoxifying, others are not. And if they are, boy, wouldn't it be great if maybe they're responding to the acetate in the media? So the idea is that extracellular acetate basically turns on some cells to detoxify.
Question?
Yes.
Just for clarification. So the acetate producing cells, are they the ones that are becoming competent? You showed a COMK.
They become competent. Yep, that's
Okay.
right.
So that's a correlation with that.
Yeah, and looking back in literature, people have done microarrays of competence, and they've seen the same genes. Nobody's ever looked at the metabolic role of this or those things, and so it was sort of buried in lists of genes from old microarrays. So in some ways, we rediscovered competence, but on the metabolic side. And so you have some cells that make acetate; these are competent cells, and then you have cells that basically will detoxify. And so we thought maybe these cells are actually, at least to some level, responding to the acetate itself. And the way we did that is we took images and we counted how many cells are expressing this over a threshold and how many are not, with different concentrations of acetate. You can see that without any acetate, just about all the cells are basically off. None of the cells are expressing the genes for acetylcholine production. As you increase the acetate percentages, you might expect that all the cells are turning it on stronger and stronger, but in reality, what you're changing is the fraction of cells that are actually detoxifying or making this, transcribing this gene, right? So this is very different than the way we typically think about the way gene expression is regulated. We think about promoter strength. We think about cells are going to turn the promoter. No, here we're saying the promoter strength is the same. Cells are living somewhere here, at least in the fluorescence unit, but more cells are active. So it's the fraction of population. With that, maybe if we make a movie, we can actually catch these cells if they are detoxifying or if there is anything like that. So we make pad movies to test this, and this is in green cells that are expressing the detoxifying gene. And you can see that later in the movie, cells that don't express it start to grow quicker. And so I'll play this again, and then we'll look at what the lineage tracing shows. As you can see that all cells are growing sort of slow, and now the cells that are not expressing this are going to start going haywire. Okay. And when we superimpose on top of this lineage tracing, you can see the expression of genes. So you can see cells that are expressing the ALSS genes are growing slowly. Cells that are not grow slowly initially because there's acid in the media, but then they start to divide more quickly and take over. And we've done this in several movies, and we show it. And it's really neat. And so then the next thing we wanted to suggest is that extracellular acetate actually turns on a fraction of the cells, and the more acetate you have, the more of these cells are on. And then these cells will basically, in some way, help detoxify the environment, or at least keep it clean. We can show this by looking at a wild type strain, and then a deletion of ALSS strain. When you add acetate, will grow, and at some point, basically fizzle out and stop growing. So at about this stage, the cells start to look pretty crummy, and then they start dying. And if you basically grow them next to each other, you can rescue this phenotype by growing it next to that guy, and they'll grow happily much further. And the idea is that maybe these guys are detoxifying the environment for the deletion strain. Okay. All right. So what I wanted to show in this first part of the talk, just using fluorescent reporters and communities, is that in clonal bacterial populations, there are especially cells that do different metabolisms. It's not just about sporulation, it's also about active, right? Because these metabolites can control this specialization themselves. So they're basically sensing the condition that they themselves create. And the most important thing is that this specialist group can actually work collectively to the benefit of the entire population in real time, not because they're betting that it's going to get worse, not because right now it's bad and someone needs to clean up the stuff, right? Okay, and so this was really exciting and it was great. Look at this picture though, say we talked about these two populations, which is great. How many populations do we actually have? Is it 3? Are all the dark cells the same? Do these green cells make one population, or are there 2 or 3 or 4? Right? I mean, these like T cells, and there's T helper and T killer and whatever the regulatory cells are, right? We don't really know this, but at the time it was becoming clear that for mammalian systems, people were able to actually do this. And we sort of, at some point, wanted to engineer a new single cell RNA-seq. In fact, reason I sort of decided to do this for the bacterial systems is that when I left academia the first time, working in aging, we took young and old mice, and we did exactly this. We basically used a mammalian single cell RNA sequencing to identify cell populations that change differently with age. And so seeing the power of this, it was very clear that something like this, if it could be done for bacteria, can really solve a lot of questions. So how does this work in mammalian systems? The way it works in most mammalian systems that rely on microfluidics is by basically using labeled oligos. So you have these hydrogel beads that basically have single-stranded oligos with a poly T on one end and a unique cell barcode. Each one of these, there's about 1 million different hydrogel flavors or colors. Each one of them differs based on this cellular barcode. These hydrogels are encapsulated into single droplets microfluidically with a single mammalian cell. And the way that these are single cell is not that each one of these gets partitioned with a single cell. The cells are basically in very low numbers through Poisson distribution. You either have one cell or no cells. And this is all done through oil. And of course, the molecular biology is pretty straightforward because you have a poly T on the end of this oligo. That poly T can bind to the poly A on mRNAs in mammalian systems. And you can basically just do very simple reverse transcription and add your cellular barcode to your mRNA and then count these bioinformatically. Okay, this is what it looks like when we're in Calico. We made some of these before. 10X and other companies made them commercial. These are those hydrogels. This is slowed down a lot. Cells come from one channel, and then the RT reactions come from another. As they move to the right, but now you'll see oil that will partition this into single droplets. Okay, so there is the hydrogels. These don't have cells in them. There's going to be one with a cell in a minute. These are mammalian cells, so they're large enough to see. There should be one with a blue arrow coming up. So you see the mammalian cell right there. And each one of those hydrogels basically has a different index on it. And so this is how the system works. It's much, much harder to do this in bacteria. Okay? Many challenges in bacteria. The first challenge is that there's no polyadenylation on the transcripts. Right? So we don't have that poly A that we can just stick our stuff on. The mRNA content is about 100-fold lower. Right? So we're looking at really, really sparse matrices. The mRNA half-life is super short. When we did it in calico with animals, I take the animal at 8 in the morning because of circadian rhythms. It was always 8 in the morning. Put it on the device at 2 in the afternoon. Transcripts were pretty stable. Your bacteria have a 5-minute half-life. You can't do that. And these systems, at least at the time, were not compatible with fixed samples. If you don't go after poly A, 95% of your transcriptome is going to be ribosomal tRNA, right, non-coding. And finally, we have the difficulty that in those droplets, what I didn't say happens, you have to actually lyse the organism. And so it's really easy to lyse mammalian cells. If you got a bacteria with a thick cell wall, you're going to have a hard time lysing it. So how do you actually get through all of these difficulties? Well, we decided not to reinvent the wheel. We decided that there's some stuff that people do that actually works really well in bacteria. And one of those, well, first of all, we can use the pre-existing microfluidics, but one of the things that people do that works really well is fish. You can actually do in situ hybridization really, really nicely. And you can count dots if you were to do colorimetric fish. And this works really nicely in single cells. Problem with fish is that you only have 5 collars, maybe 7. Maybe you can push it. Some people do combinatorial fish. I'll talk about that later. But we thought, what if instead of a collar, we put a poly A on the end of that oligo? And that way we can encapsulate it and run it through the same exact microfluidic system. And that poly A on the end of each one of these fish probes will basically tell me which cell it was in, because I have the hydrogel. So it looks a little bit like this. We take the cells, we fix them immediately, we preserve the mRNA, so we're already not worried about the half-life. We permeabilize them, so now we're not worried about the cell wall. We add our oligos that have the poly A, so now we're not worried about polyadenylated proxy for the mRNA. It's not exactly mRNA. We added a PCR handle on the other end just for easier stuff. We wash off anything that's unbound because we really only care about the transcripts, not just the probe library we made. And then we basically flow this in the same microfluidic device. And in that device, what happens is that all the polyadenylated probes in one cell will basically get a barcode. Okay. Now we can make more than one probe per target mRNA. So our bacillus had about 5,000 genes. We have 30,000 probes. And you'd think that that's really, really hard to do and very expensive. It's not, because you can buy those from Twist. It's about $2,000 and you can amplify them anytime you want to. So think about if you ordered a microarray slide, think about just amplifying the DNAF of that, use that in perpetuity. You just have to buy it once. Okay? Then basically we do the same trick bioinformatically, and this actually works really nicely, right? So this is an experiment. This is one time point in Bacillus subtilis, grown in similar conditions where the genes for acetate were expressed. And you can see that we don't just see those 2 populations or 3 populations. We see at least 10 populations here. Right? This is basically arranged by size of the population. We see the population for competence, and if you dig down lower, you'll see those genes for TCA cycle. But we see each column here is a cell. The rows are basically genes that highlight it. And I should say this is completely agnostic. I didn't tell the program which genes to look at. This is the clustering that was done by the computer. So you can see all these are competence genes. You can see here genes for SPOT, spore coat, or SPO genes for sporulation. You can see that there are a few cells that sporulate. You can see here DHBF and DHBE and DHBB. This is a siderophore that's known to be expressed. So again, like these things will get iron and other metals with a common good. This is something that's been known in the literature. But this big block here is completely new. Nobody knew that all these arginine genes, for instance, are expressed preferentially by one cell type. So it's not just one or two genes, and it's not even just one operon. There's a whole suite of genes. So we can agnostically go there without me having to make reporters. And this is one time point. We can look again at fluorescence just to make sure that we're seeing them. We can see it. The genes that are overexpressed in this competence actually breaks into two clusters. So remember I said maybe the red is like T cells and you have T killer and T helpers. You actually have that, and it's really driven by this. Therefore, you can see that the competence genes are all these green dots in here. So it really finds the entire competence. 90% of the competence genes that were known, we basically found. Sporulation, we didn't expect to have spores here, but when we looked microscopically, we sure did in our condition. So that was nice. We didn't expect it, but it was there. And you can see again, sporulation genes, not just a couple of genes. That's an arginine gene. And you can see that this is also bimodal or at least heterogeneous. And you can see the genes that are basically upregulated, and we look at arginine biosynthesis. One of the things that is really interesting, and I talked about earlier, is what are the processes that are basically segregating, and can we actually try to understand why? And the fact that we saw arginine here, we also saw arginine in E. coli. I won't show that data in other organisms. Begs the question of like why. And when we think about arginine metabolism, it's actually expressed in different parts in both plants, mammals, and now in some bacteria that we've seen. And so why are some processes heterogeneous, right? You can imagine two things. One is, or multiple things. One is there's no heterogeneity. All cells do the same. Okay, fine. We've shown that's not the case. The other thing is that in different cell types, it's completely random what is segregated. But I think what's most exciting is, are some processes really, really prone to segregation? And the reason I think that that's exciting is that that might actually teach us that some things segregate for a reason. And I go to an example from the literature in which this is really known and understood, is Anabina, which is a cyanobacteria that can fix carbon and nitrogen at the same time. And if you grow cyanobacteria where it has to actually make its own nitrogen, you see these beautiful chain-like structures where every 18 photosynthetic cells you have a nitrogen-fixing cell. And the reason that Anabina has to do this is that nitrogenase enzyme, which is super costly to make, is very sensitive to oxygen radicals. So you can fix nitrogen and carbon at the same time. You just can't do it in the same cell type, right? Or you can do it in the same cell type, but not in the same time. So some organisms have a day-night. But these two processes are chemically incompatible with each other. And what I think would be really interesting is to see whether we can actually identify other things that are incompatible by just looking at what is segregating across biology. Okay, so this basically just says what I went over. These things must be done in separate compartments. Okay, looking forward. I don't know how fast I talked. I just ran from lunch, so I don't know if I'm ahead of time or behind, but I'll maybe try to slow down a little bit.
It's 2, you have plenty of time.
Okay, so slowing down might be the way to go. So looking forward, what can we actually do with this system? What can we learn, and what are we looking at now? In bacterial system, antibiotic persistence and tolerance is a very big and hot topic, where you can basically have organisms that phenotypically switch into conditions in which they are tolerant of antibiotics. And as I showed you before, organisms can switch in and out. They can switch back into a population that's mixed. So you give an antibiotic, organisms that are usually growing quickly or basically die off, and then you have some persister cells that can repopulate the community. And so this has been shown in many different organisms. Virulence gene expression is also known to be heterogeneous in many bacteria. And so this example right here is in Clostridium difficile. It's from a lab that's now just down the hall from me, Rita Tamayo's lab. And C. difficile, what they see is that the toxins that C. diff expresses are under the control of a transposase. And so there's a switch, a phase switch mechanism that makes it so that only some of the cells are expressing. The same thing can happen for flagella or pili and happens in other places. Of course, in DuPont and other places, we look at these fermenters, and there's a lot of heterogeneity in there. So these are some pictures of yeast cells from a fermenter, and you can see that some cells are actually expressing, I can't remember what the marker was here. I think this is just for protein translation and some are not. And so you can see that quiescence can be a really big cost in these big fermenters. And so in DuPont, we really wanted to understand why some cells no longer want to produce the enzymes that we were asking them to produce. And you can imagine that that's an active area of research. As an example of what we did with some of these systems, I want to talk a little bit about Clostridium perfringens, which is a toxin. And we studied it in DuPont because it's a very important chicken pathogen. So Clostridium perfringens is the fastest growing gram-positive, actually any organism that you'll find. Doubling time is about 6 minutes, even though it's an anaerobe. And it grows very quickly, and it can be devastating to chicken flocks because it causes this disease called necrotic enteritis, which are basically like these pus-filled wounds full of the bacteria that will kill the chicken. And it does this using a toxin called NETB, which perforates cell membrane. And so very interesting organism, and it's becoming more of a problem now because chicken farming is relying less and less on antibiotics. Everything is antibiotic free. In some places, it's illegal to use antibiotics, and so we're seeing more of this organism. So we wanted to understand a little bit more about the biology of Clostridium perfringens. What we did is we took Clostridium perfringens and we grew it on rich media in which it's known to express toxin. And we clustered it again. And instead of showing you a heat map of all the different cells and genes, I'm showing you a U map, which is basically a two-dimensional representation of these different populations, in which cells that are a little bit more closely related are clustered together, although it could be sometimes slightly misleading with the space. And you can see that in some of these clusters of cells, which would have been the same clusters that we saw in the heat map, is very little toxin. It's all over here. But some clusters are really, really heavy. Almost all the cells in the cluster are producing toxin at high levels. And so this is a little bit like what people see in Clostridium difficile, where toxin really is differentially expressed. And it's the number one marker of this cluster of genes. So in the heat maps, this is a gene that goes on the top. And looking at these other populations, what we notice is that metabolically, the cells that are not producing toxin were very different in their physiological properties. And their usage of short-chain fatty acids was actually one of the big things. So again, going back to acetate and butyrate and things like we've seen before with bacillus was what was different. And so with just this one experiment in one time point, we actually had some ideas of, well, maybe if we add some of these short-chain fatty acids, we can actually affect change. And when we did that, we added acetate to the media, to the growth media. You see that there is less toxin produced. And in the main toxin producing cluster, you see it even more clearly. And that cluster itself basically shrinks. And so when we looked at this, it was very, very cool that NETB was differentially expressed. It actually turned out to be the number one most downregulated gene when you add acetate. So it's not changing its entire metabolic profile. It's just that there are less cells within this cluster, and the cells that are in this cluster are really downregulating the toxin. And so earlier we saw acetate changing the expression of a gene. And I said, this is different than the way we typically think about gene expression, because it's not just lowering the level, it's also lowering the number of cells that are actually active. Here we see both. We see it lowering the amount of transcript and shrinking the population that is doing toxin production. And we see that less toxin is also expressed. Some other stuff that we see, we decided to look at phages and how phages are induced. And so we took Bacillus subtilis, just because it's a favorite organism, and we induced phage production. So it's got a couple of phages. This is a defective prophage called PBSX. And if you add mitomycin, which basically makes cuts in the DNA, you see that a subset of cells, fairly clearly defined subset, expresses PBSX. So basically, it's activated. Interestingly enough, that's not the only phage that it has. This is SP beta prophage. The SP beta prophage is also activated when you have the same treatment. What's interesting is one or the other for the most part, right? And so why is that? And so the question is this a one or the other response, at least early on, because the type of cut and the cell state, maybe these cells are in different states in their cell cycle, they're bigger, smaller, maybe the cut was in a particular place in the genome, or what I sort of, or is it the insult itself? Or a third possibility is maybe this is a race. Maybe whichever phage gets activated first sucks up all the energy and occludes its competing phage, right? And so what is the actual biology that we see here in this kind of an environment? Okay, so now talked about what we've done, and I think where this field is heading, and lots of other cool things. Other systems of interest I'm not quite working with, but other labs are, and I think other folks will, is lab-based evolution. And we track phenotypes in real time. Can we look at how the ability to change into different fractions actually changes and is evolved? You'd think that that's an evolved trait as well, right? Can we predict which evolutionary processes will win out? And I have some ideas for that if anybody wants to talk about afterwards. Persistent intolerance, I haven't shown any work on, but in persistence, basically, the field of antibiotic tolerance and persistence is really picky about this word. The idea is that these are cells that are not translating efficiently. But can we actually start to see whether mechanisms are conserved? And then in mixed populations and communities, what happens? There's literature that suggests that there's less heterogeneity and plasticity once you put an organism with a competitor, meaning there are less niches that it sort of goes in and out of. So imagine I showed you before acetate producing cells. Well, maybe you put it next to an acetate producer, somehow Bacillus wouldn't do that. But then how does it know? How does it actually communicate? How do you decide that, wait a second, there are neighbors here? And we don't really know any of that. The last thing I wanted to talk about is the method that I developed, I think, has a lot of benefits, but there are a couple other ones that came out in the last couple of years. Ours is coming out in press in a few weeks, which is exciting. It's been in review for a long time. But other methods are also good and are very different. So the method that was developed independently by two groups, Georg Seelig at the University of Washington and Saeed Tavazoy in Colombia, is split pool sequencing of bacteria. So like us, they use fixed bacterial cells, which is good. They permeabilize them. And then they enrich the mRNA by basically selectively adding a poly A to these enzymatically. And then they do 3 rounds of split pool barcoding, which is basically adding barcodes to individual cells in a very manual way. And then they sequence those. They can profile thousands of cells, but this is very costly. Even though they try to enrich, they still see over 90% of their reads are ribosomal. It's really hard for every sample. You have days of work, and it's low sensitivity. They see 20 or so transcripts per cell. We see about 300 in similar conditions. But the main benefit is they don't need any a priori knowledge. They don't need to make probe sets like we do, right? So they do have that benefit. Some people have sorted bacteria into 96-well plates and have done things that way. This is very time-consuming, low throughput, about 200 cells. They still get 97% of their transcripts are ribosomal, but they do get very good coverage like we do. Plus, they get it throughout the entire transcript, not just where they're probes. So that's where you might want to use that. And then finally, people are using a fish-like approach that allows you to do spatial imaging. So there's marefish and seqfish, which are 2 techniques. Seqfish is the only one that's been applied to bacteria before. What you do is you basically permeabilize bacteria, you put on probes for your gene, and you do multiple rounds of hybridization with the same gene and try to basically have a color code, if you will, for the transcript. So you do one round, it's green, the next round it's red, et cetera, et cetera. And so in this paper, they did 105 genes, and they did 40 rounds of fish strip and hybridization. And each time your transcript has to be in exactly the same place. You have to segment the cell perfectly, and you have to be able to pick it. And the transcript cannot miss any of those rounds. And so this is really, really hard to do, but it's amazing, and you get spatial resolution. So there are other ways of doing it, but it's very, very hard. And I think I did talk really quickly because that's my last slide.
Thank you, Adam. Times for questions. Orlando.
The single cell methods are really amazing, as usual. But I actually have a question about the arginine producing bacillus cells. Because arginine is a very special amino acid, right? It could act as a chemotropic salt, affecting the solubility of other proteins. So are there any ideas of where these cells are producing arginine to control the solubility of the cytoplasm or surrounding cells or something along those lines?
So arginine can be lots of different things, like polyamine synthesis. It can be lots of different things that it does. I have no idea why those cells are doing it. We don't have any movies of that. And so this was something that we found when I was in industry. And despite my pleading, they were not happy with me spending time trying to find out why arginine, because the cells were still producing enzyme and they just didn't care. And so, but I would love to know why arginine. We see arginine now in 3 organisms that we've done careful stuff in minimal media. We obviously don't see some of these things in very rich media. But it is something that we see in multiple bacterial strains, but I have no idea why. And I'd love to, if there's some ideas of cheotrophic.
It would be interesting if it induces dormancy in other cells. But
I mean, in a movie, you'd be able to see whether that turns on and then those cells are dormant. And so, yeah.
Gavin?
How would you distinguish between like different populations of cells doing different things and like a slow periodicity in metabolism where all the cells are doing the same thing, but just at different phases of that periodicity?
Yeah, so that's sort of goes to the dynamics of processes. And that's where it's really hard because we're just seeing it in a snapshot. And especially in bacteria, transcription is really bursty. And so are we just looking at, oh, look, these 2 operons are just there because for whatever reason that was stochastic and that's why these cluster. And this is, again, like where movies and follow-up really go a long way. And so I think that the idea is first you find out what's heterogeneous, and then you find out what's long enough lived and biologically relevant and is not just a switch. I think hopefully that answered the question.
Because like in yeast cells, metabolism is periodic. It's like they go between 2 different states over, I don't know what the time period is, but I don't know if that's longer or shorter than the cell cycle.
Yeah, so in yeast, there's also like the metabolic oscillations that people see. I don't know how well people see them in cells that are not synchronized. I know that if you have a chemostat, they will synchronize, and the idea is that this really is a protective effect, right? Because the correlation of when they are aerobic is opposite to when they're actually synthesizing DNA. So the idea is you're protecting your DNA from oxygen radicals. You're going to ferment, and then you're going to grow fast and do. I don't even know if in yeast, and Nick, you may know this better, if this is truly something that people see. But even in bacteria, like, right, if this is just a switch between states, is the order always the same? I don't know. Even more than this, if you look at cell cycle in bacteria, how much of the transcriptome is really cell cycle control? People haven't done these experiments.
Yeah, well, I'll just follow up on the yeast thing, but if you want to, also, if you click on the chat, I think there might be some questions there for you. And I'll just say, yeah, with yeast, people have looked at single cells not synchronized in a chemostat, and they will see that there are metabolic oscillations. If you look at things like using fluorescent reporters to look at certain compounds, or if you look at autofluorescence, which is usually NADH and whatnot, they'll see oscillations and connections to the cell cycle.
And there's a nice explanation of why, which is a really lovely idea that, okay, there's a reason why these things are kept that way. In bacteria, a lot of times the synthesis of DNA is throughout the entire, right? They don't really stop. And so the question is, well, if you're going to respire, you don't have the luxury of stopping, but maybe we do see it in higher resolution, some differences. So questions, where can we do bacterial single cell RNA-seq commercial? So that's a good question. And then the second one is, in parallel to SR, could you capture live single cells for phenotypic testing? Okay, so I'll answer the first one first. What I've developed, we now use a 10x device, which I'm sure there are plenty of folks in NC State. If not, we're down the street. You can come and use the 10x microfluidic device. What you need to do this is you need a probe set. And so I have probe sets for some organisms, which I'm happy to share, but some of them you need to actually create those on Twist. And I'm happy to send out protocols. At some point, I've just started my lab, and at some point, the department at UNC asked about the possibility of starting a core facility that will do some of this stuff. I'm not sure if I will or not, but that's something that may happen at some stage, in which case you guys are obviously welcome to. But the main thing is getting probes. If you can do fish on your organism and your organism doesn't chain into something that cannot be broken, this generally works. And the second one was about full circle capturing live single cells. So we've tried to do some hysteresis type work by sorting on fluorescence. In fact, sorting on fluorescence is something that we thought before any of these techniques would be the way to go. The problem when you sort on fluorescence is that fluorescence is fluorescent GFP. Your fluorophores are a stable molecule. So by the time you sort on it, if it's a fast dynamic, you're way too late. So I showed you guys that we can recapture competence, and we were somewhat lucky. We tried to do that for the acetylcholine-producing population. We never got it. And if you look at the movies, they just turn on and off very quickly. So by the time you're at peak fluorescence, you're probably not transcribing it anymore. And so that's why, like for some things, it's great to be able to go full circle and sort, but it really depends on what the phenotype is and the dynamics of that.
I think that may be the only question. Any questions in here?
Yeah, so you've made reference to types, but also states, and these are clonal populations. So I wonder if you can expand a little bit more on whether you think there's a role for like genetic differences even within those clonal populations that are specializing, as you mentioned, or if it's just really just exactly same genetic material leading to different states.
Yeah, no, so that's a great, right? These are all like human terms to describe a state. And the reason that I'm somewhat loose with the language, I think, is that the language has not really been well clearly defined. Certainly for some changes that we see, like toxin production in Clostridium difficile, which Rita Tamayo sees, you have transposition, which is a genetic program. And so some of those are. Competence is not, it's a protein. And so I think that you see both of these. And I think what's really an interesting part of what we have to do better is to try to understand when do bacteria use one of these mechanisms, when do they use the other one? Because even the transposition, you can transpose back and there are different rates of transposition. But these are generally more long-lived versus some protein, right? And so this sort of asks the question, on a cellular level, are there different ways to encode memory, cellular memory? And you can imagine that different strategies, whether they are truly cell states or cell types, which are genetic, are that way. And then that leads us to the next thing, which is real changes that don't go back, like evolution. And so if you have an evolved population, can you now start to see, okay, what's going on? And that evolved population might have a genetic change, but you may have cells that are still in those different states, but just with a different genotype. So that becomes more difficult because there are those two cells that are on incompetence, but are genetically distinguished. Are they in the same state? Are they the same cell type? Are they the same, right? But I think that the language is still really loose right now. And,
Just to follow up on that, if it's that stage where there is the genetic differentiation, you're talking about, in some of these cases, a mutualism.
yes,
Right? And once you start talking about a mutualism, you mentioned DuPont, and they're wondering why isn't the bacteria producing what it's supposed to, right? So there's a cheater kind of dynamic going on. So how that's controlled and not having cheaters in the system seems to also be the question. If it's real differentiation, are you thinking about?
Yeah, so, the classic cheater, one of the classic things is in processes that produce public goods. And so one of those is siderophores. So I showed you siderophores are bimodal. Interestingly enough, what we see is the cells that are producing the siderophore are also producing the uptake machinery. And so it looks like somehow they're organized not to cheat. That doesn't always happen.
Yeah.
What happens when the system breaks? And I'm sure that there is some cheating and there's some way to regulate them. I tried to pitch that idea to DuPont unsuccessfully. But I do think that we can start to learn that by actually looking at evolved populations where cheaters actually can crash the system, or by mixing things. Other things that you can start to look at when you have evolved populations is if you evolve on one stress, let's say I'm E. coli and you put me under acid stress, what I'm probably going to evolve is the general sigma factor. And there are going to be a whole bunch of mutants, either in sigma factor or other things, that are going to have more or less fitness. If I let it play out for 3 weeks, one of these is going to win. The one that's best is going to win. Other ones that were a little bit better than wild type are not going to win, but they're there early on. Yeah. Can you do this now? Look at the single cell population and predict which ones would now be better if you add salt, or if you added heat, or if you added. And the reason I say that is you can imagine that with an antibiotic course, maybe in a 3-week antibiotic course, select the best one. But in reality, earlier on, if you switch to a second antibiotic, there are a whole bunch of other ones that are there that are dangerous. And so it's sort of like cheating. There are lots and lots of community effects where you have genetic diversity, which I think would be really interesting. But we haven't done any of that because it's really hard to actually then look at what the genotype is of those single cells.
Yeah. But it seems like you'd be able to, in some sense, make your own synthetic communities in some ways and watch the dynamic of who's next to who and who succeeds and who doesn't.
Yeah, exactly. And really, if they're competing in a certain way, are there populations that now shrink? And are they? So we haven't done any of that, but there's a lot of fun stuff that can be toy models and things like that.
Yeah, I had a follow-up question to the arginine story.
Yeah.
If I understood it correctly, right, these bacteria are grown in a medium that had plenty of arginine.
No, this isn't. So we do our stuff in M9 media. So it's really synthetic media. And so they actually have to create it. Although, and so that's one of the differences in some of these other methods. People use rich LB media, et cetera, and they see lots and lots of transcripts, little phenotypic heterogeneity. The poorer the media, the more we see. You basically have to make the cells work.
That means that the other cells have to get the arginine. I mean, is there like transporters also?
So it's not clear how other cells do. Other cells spend some time of their life there and then store it up, or do they actually transport it? In Anabina, you see those photosynthetic cells and then the nitrogen fixtures. The nitrogen fixture will basically export glutamine, and as that gets really, really low, the next cell in line will be like, wait, there's not enough glutamine. It will make glutamine, and that will be exported. Here, you'd have to do it extracellularly in a different way because you're not in a big chain. And so it's a little different, but I don't actually know. Like, are these cells making so much of it because they're making polyamines, and other cells are still doing it? I don't know.
Do you see a pattern in the uptake transporter that is counter to the pattern you see, or is it?
I'm sorry, I didn't.
Do you see like an uptake transporter for arginine in the other cells at all?
Oh, I'd have to look back and see. I don't remember actually looking at only this population versus the entirety, and that might be missed. There was nothing that jumped at me. But it could be. Interestingly enough, with the arginine story, I mentioned that in eukaryotes, it's also segregated. What's segregated is actually half and half of the reaction. So half of the reaction happens in the cytoplasm, half in the mitochondria, and it goes in the same step where it's cut. Here, we don't see that. We see all the genes in there. And so I don't actually know. It could be that what's transported is ornithine, and cells can do part of it. Here, I don't think they do it because the entire process is there. How much arginine does a normal cell need? Could be that basal levels are fine, but if you're a specialist making something, I had very hard time getting traction on arginine in industry. And looking at toxins is easier to fund, so that's why. But I think that as we see it in more and more organisms, I do hope to follow up, because that is a case in which there's a clear incompatibility, or I don't know if incompatibility, but there's a chance. Yeah.
I think, Eric, I saw you, and then
Just a bit of a technical or process question.
Yeah.
Different growth conditions, different media, sorting, et cetera. And I was thinking of permeabilization. How do you permeabilize the cells to get those probes in? Is that changing much in your protocol, or you worked it out for gram negatives, positives, mycobacteria?
So we use formaldehyde fixation, which is about as immediate as fixation as you can get. And so that's the best that we can do.
They're dead. Cells are
They're dead. They're dead, and the entire fish field is based on that.
Right.
But we don't see differences between gram positives and gram negatives in terms of that. When we start to do this in tissues, I don't know how quickly it will diffuse through if it's cells that are in a lung or something. So that may be a trick. But that was a
Last question, Jessica.
Yeah, I was just really curious watching your microscopy videos if you've noticed anything about maybe say a theoretical sphere of influence that a single cell could have, because a lot of them seem to have contact with each other, but I could also think you could make the argument that maybe they're influencing other cells based off of like a diffusion rate around them if it's a environmental factor that's causing these changes. Have you noticed, is it like strictly like the changes you see are happening like cell to cell neighbors, or is it more of like a general area around the cell? Like,
yeah. So like the spatial type arrangement, we did two different types of movies. So one movie is the Mother Machine, in which we saw no spatial stuff. It was purely stochastic. As people talk about competence, it was that way. And what we saw is that if you change the media, the conditioned media that you put, you get more or less competence. But where it happened was random and didn't change with different channels or with it along the channel, et cetera. And it wasn't influenced by what the mother cell was, right? So daughters weren't more likely or less likely. In the movie in the PAD movie, you definitely see spatial arrangements where cells that are closer to the center appear to be more active or fluorescent, et cetera. It's really, really hard to then actually influence that, because if you have a movie like that, if you ablate the cell, now you have a lot of dead nutrients. And I don't really know how, so we weren't able to really ask that. But I definitely think that there are spatial components that drive this. And looking at spheres of influence is something that with lots more movies could probably be done. We didn't do it here, but I think the spatial component is really interesting.
All right, let's join me in thanking Adam for a great talk.